And let's say it gets to 50% and 50%. So here we have gas moving in this direction, and then it stops, right? So even if there's like 50% saturation in the water, it will not diffuse because it's equal to what is in the blood. And so that's why it's really important that countercurrent exchange will allow the fish to extract more oxygen from the water. So even when the water has only 15% oxygen in it, diffusion will still take place, okay? So it still occurs. Does that make sense to everybody? So, engineers have done this with water, where they will like actually take and run pipes. So you have your drain in your shower and it's hot water going down the hole, and they're like, that's a lot of wasted heat, the water going down the hole. So what sometimes they do in like eco buildings is they'll put the water pipes next to the drain, and heat from the drained water will actually go into the hot water and will actually keep the water warmer, right? So you don't lose the heat. And so sometimes they build heat exchangers in the same way. And actually, we'll talk about this, but this would be the way that birds keep their, like ducks, keep their skinny feet warm, is because of countercurrent exchange. Okay, so that is a adaptation, a special adaptation by fish. So in your book, they show this, right? So water is coming this way, blood is flowing counter to that. Okay, so amphibians and reptiles, remember that amphibians have the ability to breathe across the surface of their skin. So their lungs don't have a lot of surface area. So they don't have a big surface area. So they use, so this is specifically amphibians, they use the surface of their skin for respiration. So this is why frogs and salamanders have thin, moist skin, but it's not very protective, right? So they lose water across the surface of their body, so they dehydrate. So even if you have a toad living in the desert, they generally have to kind of go underground to prevent dehydration, and then they come back up when the rains come in the desert in the spring, and they mate, and then they kind of go back down. So you have these toads that kind of live either in springs or underground to prevent dehydration. Okay. So what we see with the reptiles is that the reptiles have scales. So they cannot breathe through their skin, but the scales prevent water loss. So reptiles can be more terrestrial than amphibians, but they have an increase in the surface area of the lungs. Now, the interesting thing about these organisms is that they have to force air into their lungs by gulping it. And so sometimes you see that their throat muscles kind of forcing air into their lungs. And so these organisms lack a diaphragm, which we're going to talk about the significance of in a minute. So they have no diaphragm, and they have what is called positive pressure breathing. So they have to force air into their lungs, which we thankfully do not have to do. Okay. Positive pressure breathing. So they force air into lungs. So when we look at the evolution from amphibians to reptiles, remember that the fish have a primitive lung, which is the swim bladder. So the swim bladder, you can see here, is just an out-pocketing, right, of the pharynx. So this shows bony fish. That's what the lung looks like, right? And then if you look at the amphibians, this becomes modified. So in some amphibians, they have a greater surface area. but it's still connected, right, to the pharynx. And then if we look at the difference between amphibians and reptiles, here's the amphibian lung. It doesn't have as much surface and it's not as large as the reptilian. And then when you look at the mammal lung, it has a lot of surface. And so the reason why we need to have a greater surface area is we need more oxygen. And why do we need more oxygen? Nope, not necessarily. Thinking like a shark, or we're thinking, there's some big reptiles, not big. There's dinosaurs. Yes, so we need oxygen to regulate body temperature. So we need to obtain more oxygen so we have a greater surface area, right? Because we are endothermic. So this is an adaptation to endothermy. It's having a big surface area in our lungs. Okay, so let's talk about respiration in birds because they are also endothermic, right? So these are endothermic, so they need to have a very efficient mechanism. So they also lack, because they're reptiles, right? They lack a diaphragm, but they have a lung called a parabronchi. So they have parabronchi that allow for the one-way flow of air through the lung. Allow for the one-way flow of air through lung. Okay, so this is an adaptation that maintains the delta P of like oxygen, right? So if you think about it, our lungs are kind of weird in that air goes down in them and then it can sit there, right? And you can have what they call dead air. And dead air is where there's lots of CO2. And so if we don't breathe deeply, sometimes we can be oxygen deprived, right? So we breathe deeply, we get more of the surface ventilated, right? And so birds do not have this problem in that they do not have a sac where their respiratory membrane is maintained. So they also have air sacs. So they also have air sacs. Okay. And the air sacs have a couple of functions. They are functioned in buoyancy, so they make it light, so they lighten the bird. So birds tend to be really light because they're filled with air, right? And it also stores air. So I would say that the birds have a little bit more efficient mechanism of gas exchange than we do. Okay. So if we look at a diagram that shows this movement of the air, okay, the total, in order to get the air in and out, they actually have to inhale and expire twice, 2 cycles. So where we inhale the air and then we can exhale the air, in order to get the air to move through their lung, they have to do it twice. Okay. So if we look at the movement of the air, it comes in and then it gets put into these posterior air sacs, and that expands, and that makes them light and buoyant. And then on its exhalation, the air then moves through the lungs in one direction, right here. And then it gets stored in this anterior air sacs, and then upon inhalation again, the air will move out, okay? So we have no dead air in the lungs, okay, which means that we have a more efficient diffusion of oxygen and CO2 between their circulatory system and their respiratory system. Okay. So respiration in mammals. So I want to talk about ventilation. So this is the movement of gas over the surface of the respiratory membrane. I'll just put air over surface, okay, of alveoli. So how do we ventilate? We have a very special mechanism for ventilation. It's not seen by any other organisms, and it's called negative pressure ventilation. And we do not have to force air into our lungs by gulping it. So it's not a physical exertion to get air into our lungs. Okay. So this is due to the diaphragm. So if I drew my trachea here, and these would be my bronchi coming out to my lungs, okay, and these would be my lungs, okay. There is a muscle that sits under the lungs that is called the diaphragm, and it is kind of an interesting muscle because normally we don't think about contracting it, but we can cause it to contract when we breathe, so it tends to be involuntary. Okay, so that's the diaphragm. So when we look at the diaphragm, its orientation is really important because it affects the volume and the pressure of air in the lungs. So when it is relaxed, it is dome-shaped. And it presses against the lungs. Pressing against the lungs, it decreases, so I'll just write some lines here, decreases volume in the lungs. And this increases the pressure. Okay, so just like, you know, if you decrease the volume of a balloon by trying to squeeze it and make it smaller, the air in your balloon will exert more pressure. Right, and it'll be likely to pop. But in this case, we have a valve where air can move out, right? So in this particular instance, this would be exhalation or breathing out. Okay, so pressure, this increase in pressure forces air out of the lungs. So that's when the diaphragm is relaxed. Now, sometimes we think when we exhale deeply, it's kind of like, well, how come it feels like I'm contracting stuff? You know, when I'm like, oh, forcing air out of my lungs. But we're actually contracting our rib cage and our abdominal muscles. So if I want to force a lot of air out of my lungs, I contract my intercostal muscles, which make my whole rib cage press in on my lungs, and that decreases the volume, and that makes more air move out, right? So exhalation normally is not something that is doesn't take a lot of energy because the diaphragm is relaxed. Okay, so when it is contracted, the diaphragm is contracted. Okay, the diaphragm flattens out and moves down. So what this does is this increases the volume of lungs, right? And that would decrease the pressure. This is the negative pressure, right, that they all talk about, right? Negative pressure breathing. Why do we have negative pressure breathing? Because when the diaphragm contracts, it makes the lungs expand, and then all of a sudden there's like a vacuum. And so air will just automatically flow in, right? So this is inhalation. And so you're like, okay, that makes sense, but it doesn't really make sense until you sit down and think about it, because it's really easy to get this confused, right? It's like, oh, it's really, there's lots of variables there that you have to think about, okay? So the diaphragm contracts, that's when we inhale. When it relaxes, that's when we exhale. So in your book, they have an image of that. Okay, so this is the diaphragm again. They also talk a little bit about, in your book, the role of the rib cage in inhalation and exhalation. Okay. So the diaphragm contracts and moves down, and air just automatically flows into our lungs. So that would be the negative pressure exhalation. We would contract our intercostal, so the ribcage gets smaller, and the diaphragm would move up. Okay. So I think we're going to take a little break here. So we'll come back at like 1 o'clock, and we'll have a little bit more lecture. So you can get up and walk around and use the restroom. disk. Okay. So when we talk about the movement of oxygen, remember that oxygen has to move from the atmosphere, across the respiratory system into the circulatory system. And then it has to be passed off into the cells, which are surrounded by interstitial fluid. So we're going to talk about how oxygen is transported, and in our blood, we have a special type of blood cell, which is the red blood cell. And these are also called erythrocytes. So those are the red blood cells, erythrocytes. And you can distinguish them from the white blood cells, which are called leukocytes, which we'll talk about in a little bit. So when we look at the red blood cells, they have a respiratory pigment called hemoglobin. So this is our respiratory pigment. And this is what actually gives our blood its color. And interestingly, it never is blue. So when we look at it and it looks like our veins look blue, that's because of the light moving through our skin. But it's either dark red or light red. So it's dark red when it's unoxygenated and more light red when it's oxygenated, okay? So, but we use blue and red just because it's convenient on diagrams, okay? So the hemoglobin is a molecule that consists of 4 globins, and these are proteins, and 4 heme groups. And the heme groups have a mineral in them. And what is the mineral in the heme group? Does anybody know? What do you have to have? What mineral do you have to have in order to produce blood?
Calcium.
Not calcium, iron, right. So we use iron. Now, I want to point out that insects use copper, right? So some invertebrates use other minerals to transport oxygen, but we happen to use iron, okay? So if we look at this model, it is the iron part of the heme group that binds oxygen. So the iron binds oxygen reversibly. So what this means is that it picks up oxygen and then it releases oxygen where it's needed. And this is a super important characteristic. This is the same molecule behaving dramatically different in different parts of the body. So out in the muscles, which are using oxygen, right, it'll deliver it and it'll release the oxygen, whereas in the lungs, it picks it up. So it's a kind of a special molecule. So when we talk about hemoglobin saturation, which I actually mentioned earlier when we were talking about saturation, this is the fact that 4 molecules of oxygen can bind to each hemoglobin molecule. Each hemoglobin molecule. So if it is 100% saturated, that means that all of those binding sites have oxygen bound to them. If it's only 50%, that means that 50% of the sites, right? So we can talk about saturation and hemoglobin, okay? So if it's 100%, that means it has all 4 molecules attached to it. So if you look at a picture in your book that shows this, these are the red blood cells. They have a special shape to them. And another really interesting thing about our red blood cells, mammals, the red blood cells of mammals have lost their nuclei, and that actually allows them to pack more hemoglobin into their red blood cells so that they can transport more oxygen. So if you were to look at the blood of a fish or an amphibian or a reptile, it actually has a darkly staining nucleus in it. And they don't need to have a lot of hemoglobin because they're not using oxygen to produce heat, okay? So this is the heme part, these are the iron, and then we have the binding of the oxygen to the different parts of the hemoglobin. These right here are proteins, okay? Okay, so we can talk about hemoglobin dissociation. So dissociation, let's see if I can spell that right. Let me just get it. I can't ever remember how to spell that word. Okay. Oh, it's not on here anyway. Okay. Dissociation. Ah, sorry.
No, you had it right.
I did have it right?
Disassociate.
I know, but it's spelled weird. I think, let me just look it up. Dissociation. It's not it's dissociation. Dissociation. There's 2 S's and there's an O. Okay, I knew it was. Okay, this is a weird word. 2 S's and an O. Okay, dissociation curves. Okay. So these describe when oxygen will be taken up and when it will be released by hemoglobin. So when hemoglobin will bind oxygen and when it will release it. Okay. So hemoglobin behaves differently in different environmental conditions, and the environmental condition that's looked at here is the partial pressure of oxygen. So you notice that the partial pressure of oxygen is just the concentration of oxygen in the blood, right? So and also in the interstitial fluid. So here you can see that it's very low. Here it would be high. And so you notice that that hemoglobin will pick it up when PO2 is high, but when PO2 decreases, right, partial pressure is low, that means the oxygen is being used by metabolically active tissues and it's being removed. And so you see that there's a dramatic decrease in the saturation of hemoglobin. So you could write this as you increase the partial pressure of oxygen, right, you will decrease saturation. And this would be in the tissues. We'll just write tissues. Okay. Oh, sorry. Just should be decrease. Sorry, decrease. Okay. As we increase the partial pressure, we increase saturation. So this would be in the lungs. Now, there's a couple of other things that affect dissociation, and one is temperature. So when we look at tissues that are using a lot of oxygen, they increase their temperature. So as we increase temperature, oxygen is released, right? So we would decrease saturation. And then also pH plays a role, right? So pH is a measure of acidity. So if we decrease the pH, then that makes it more acidic. Okay. And so this would mean that as we decrease the pH, more O2 is released. So we increase we decrease saturation, decrease saturation. Two different ways of saying it. Okay. Now this pH is actually due to the presence of carbon dioxide, because you remember when we talked about ocean acidification, when carbon dioxide mixes with water, it produces carbonic acid, right, which then releases hydrogen ions and carboxyl ions. Okay, so this also has to do with the partial pressure of CO2. So if you have more CO2 and metabolically active, that's going to decrease the pH. So I can also write, as we increase CO2, we're going to decrease pH, and we're going to decrease hemoglobin saturation. Okay, so that's the reason why pH values differ in different parts of the body in the blood. So this is blood pH. Okay, so that is happening in your blood. Okay, so we also transport carbon dioxide as well. Okay, and so in the blood, so carbon dioxide, so this just shows how, if we look at the percent of hemoglobin saturated, notice that as PCO2 increases, saturation would decrease. Okay, so that relationship between those two values. Okay, so let's look at how we transport CO2. So the majority of CO2 is transported as bicarbonate ions. So this would be HCO3. Okay, and this is in the blood plasma. So I think that that's like 70% of it is transported like this. Okay. And then we have a small part that is just transported dissolved. So CO2 is just dissolved in blood plasma. Oops. And so that's like 7% of the carbon dioxide. And then interestingly, CO2 can bind to the protein part of hemoglobin. And so like 23% is transported in that fashion. Okay. So it doesn't compete for the same binding sites as oxygen. It binds to a completely different part of the hemoglobin molecule. So this just tells us that hemoglobin does play a role in transporting CO2 as well as oxygen, but it's not as important, right? Because CO2 can be transported in with other mechanisms. Okay, so we're going to start with the circulatory system today. Okay, so this is another chapter in your book. And I just want to point out that we've talked about the difference between closed and open. So the earthworm, for example, has a closed circulatory system where the blood is always contained within vessels. Whereas when we look at the circulatory system, say the crayfish or the clam, that was an open system because the fluid that contains the oxygen and nutrients just simply goes into a body cavity and then into the tissues and then has to make its way back up to the heart. And so here you can see that this is the way that honeybee, for example, would circulate its blood. It's not always contained within vessels. Sometimes it goes out to a central body cavity and percolates through the tissues. Okay. Oops, we already talked about blood. So. Okay. So let's talk about vertebrate circulation. So we have a closed circulatory system. When we look at the other vertebrates, fish, for example, have a two-chambered heart. And this is kind of makes sense when you look at the way that their circulatory system works. So we have a chamber that receives the blood, and then we have a chamber that pumps the blood. And blood comes in this direction and out this direction. Okay. So what is the name of the chamber that receives the blood? Does anybody remember? Ventricle. Nope, that's the opposite. What's the other one besides the ventricle called? Atria. So this is the atria. So if blood is flowing into the heart in this direction, that's what receives the blood. Okay. And then the ventricle is what pumps it. And then blood goes out to the gills, then out to the body, and then back to the heart. So the thing about this system is it is a single circuit. And that is why we only need one atria. Now, it's not very efficient because once it goes out to the gills, it starts to lose pressure as it goes through the capillaries, and then it's under low pressure as it goes out to the body. So blood starts to become really slow moving by the time it gets back to the heart. Right? So if we look at the amphibians and the reptiles, they have a three-chambered heart. So in this case, they have two atria and one ventricle. And they have a double circuit circulation. So it's two circuits. So I'm not gonna draw the heart. I'll show you in a picture. You have one in your book. So the heart, blood goes out to the respiratory system. Then it goes back to the heart. Then it goes back out to the body and back to the heart. Okay. So you can see here that it returns to the heart twice. So this is what is referred to as the pulmonary circuit, if it's the lung, right? So I'll put pulmonary. And this is the body, so it's called the systemic circuit. So the problem with having only three chambers, however, is that you get a mixing of oxygenated and unoxygenated blood. So you notice here that if it's coming back from the lungs or the skin or wherever it's picking up oxygen, it is going to be oxygenated, right? If it's coming back from the body, it's going to be unoxygenated. So we do not have complete separation. Of O2 poor and O2 rich blood. Okay, so no complete separation. Okay, so the endotherms, right? In endotherms, including fish and mammal, or not fish, but birds and mammals, right? We see that there is complete separation, okay? So this is my fish heart, this is my amphibian heart, and notice that they have purple here. And that just shows us that there's mixing of oxygen poor and oxygen rich blood. And then if we look at reptiles, it's kind of like an intermediate. It's a transition. There's almost a complete separation, but there's a little bit of purple showing there. And then if you look at birds and mammals, they have a 4-chambered heart. And so we see here that they have complete separation of oxygen poor blood coming from the body and oxygen rich blood coming from the lungs. And so mammals and birds, the endothermic adaptation, we have 4 chambers. 2 atria, 2 ventricles. And complete separation. Right, separation of oxygen rich and oxygen poor blood. Okay, so I'm going to show you a video to end around this class. And this video is, I love this video because it actually shows a heart in a person where they're doing surgery. And it also talks about the problems when your heart stops working properly and how they actually have to go in and put a pump into your body that would normally do the work of specifically the left ventricle. So the left ventricle typically is bigger and stronger because it is pumping blood out to the body rather than just out to the lungs. Okay. So this is the last thing we'll do for today.
Montefiore Medical Center. With just 7 hours left before he drops off the priority list for a transplant, Bob Cunningham has finally gotten the call. A donor has been found, and this time the prospects are strong.
The donor team just left. Okay. Ready?
Oh, yeah.
This is it. Ready, whoever be. And we're ready.
With time of the essence, 2 of Montefiore's surgeons set out to bring back Bob's new heart. The donor, a 38-year-old victim of a car crash in North Carolina.
We are referred to as the harvest team, which is sort of a euphemism for the team that's going out to get the organs.
The team must fly to North Carolina, remove the donor heart, and return to New York, a distance of more than 1,000 miles.
That's the airport.
How are you?
Fine. Wires. Literally.
I had to break this party up, but we really got to get going up the hour.
Fine.
Very much. You better be good.
I'll be back in a while. Everything's gonna be fine.
Within about 5 or 10 minutes, they're going to start anesthetizing him, putting lines in and everything. We want to be ready to cut skin by 7:30.
Not a perfect landing.
Everything depends on whether the donor heart is healthy. The Harvest team in North Carolina examines the heart before signaling doctors in New York to begin surgery. The two surgical teams must coordinate their work precisely.
So we just got a phone call from the Harvest team. They've looked at the donor heart, and the heart looks great. So we're going ahead and proceed. The first thing is you're going to go back through his old incision, just open the same incision he's had, and then we have to deal with scar tissue. And it's very unpredictable, but that's going to be easier or hard. Remember, he's had 2 prior operations. So it can be quite difficult to start digging to free up the heart. But we're going to get started. Incision? Thank you.
In North Carolina, the critical moment has come. Bob Cunningham's new heart is removed and put on ice. Without blood to feed it oxygen and nutrients, the heart muscle cannot survive for long. The team has just 4 hours to get the heart back to New York and into Bob's chest.
We got very serious bleeding up on the chest. We need blood in the room. We got into some troublesome bleeding opening the chest. No, stop. We got a lot of air. Okay. Yeah, we're gonna have to circle rest.
Unfortunately for us, something bad happened right after the surgery started. You know, we cut skin and everything was fine, and we cut sternum, cut bone, and with all of the bleeding, it turned into, you know, a controlled emergency.
We're getting on the plane. It's probably an hour and 15 minute flight, and then about a 30-minute ground time. Can you guys go back and stop the light though? Give me what I get.
As the surgeon tries to cut through the layers of scar tissue encasing Bob's heart, the bleeding grows out of control, making it impossible to see. With time ticking away, he takes drastic action. He orders all the blood in Bob's body removed and replaced with ice water, dropping his temperature to 64 degrees, slowing his metabolism and diminishing his need for oxygen. Bob is put into a state of suspended animation.
He's got no vital signs. I mean, if you look up at the monitor, he's got no EKG rhythm, no arterial, no blood pressure. So for the last 30 minutes, he's been pretty much clinically dead. We've drained all the fluid from the field, and he can actually work now to get through the scar tissue to visualize everything that they need to visualize.
You guys mind calling Mentor? It'd be good to have an extra pair of hands. Wow, completely stuck here, huh? Unbelievable. Where's this heart? I can't even tell where the heart is.
Doctors have passed the point of no return. The surgery has damaged Bob's heart beyond repair. But they still have to cut through more layers of scar tissue before the new heart arrives from North Carolina.
We just landed. We'll be there in about half an hour. Bye.
Some semblance of a heart here. Okay, let's see if I can find the order here. Now let's get the left atrium cut, and then I'll be over. All right, here we go. I think you're going to be good now.
With Bob still in a state of suspended animation, the doctors slowly return his blood to his body and raise his temperature back to normal. And now Bob's 63-year-old heart, diseased, damaged, and still attached to the heart pump that has kept it beating for the last 2 months, is finally removed.
There it is.
Just as the donor heart arrives from North Carolina. Over the next hour, doctors will sew Bob's new heart into place. They must connect 4 vessels and let Bob's own blood flow into the transplant. Success will be measured by a single muscle contraction, the first beat of Bob's new heart.
Here we go. Here comes the clamp off. 0.5? 0.5 in the top. Okay, clamp is off. Paddles with us. Well, again. Okay, good. We have initial beat.
I've done over 150 heart transplants. It's one of the toughest ones, for sure. I think he's going to do well. The only issue we're going to watch very closely is how does he wake up from the operation. Despite all the difficulties and challenges, I think all will be okay.
So that heart that they took out of him was attached to a pump, and that's sometimes referred to as the LVA assist, so it's a left ventricle assist, and that's actually connected to a battery supply that's outside, and that cannot be sustained, or cannot sustain the body for that long, right? So that's why they would have to do eventually a transplant. Yes.
So like the big metal piece they pull out.
That was inside.
Yeah, where does that sit? Like where do
It
they
must
get
be,
that?
you know, it sits in there. It fits. I'm not sure. That big metal thing, that was the mechanical pump that was actually doing the work. So I bet it felt like his ribs were out to here.
Yeah, probably expanded his ribs. Right?
Yeah.
Okay.
Yeah. Yep.
That's crazy. Okay, that's cool. That's awesome.
Okay, so we will. Or I will see you tomorrow for that. Also, the cadaver, we want to think about whether or not we want to participate in the cadaver moving.
I just know you don't. That is still tomorrow.
That's tomorrow.
Okay. That's crazy. So when you were talking about the diaphragm, is it all humans that have it or mammals?
Mammals.
All mammals have the diaphragm.
Mammals have it. Yep. Yep. Yep.
I have a negative blood, so I feel like I should donate blood. But I heard somewhere that like if you donate during a blood drive, a lot of that blood gets like tossed out because it gets too old or something like that. Like, is that true? Do you know?
I don't know. It probably just depends upon like how fast it's being used by the county. So I don't know.
Okay. And
I'm
do you
not
know
the
like
person to
if
ask
I
them.
can just like give blood whenever? Or
You could call the Umatilla Health County and see if you can just go in and donate blood. So it would be during a blood drive.
Okay.
Yeah,
Thank
yeah.